So key things to remember about proteins. First, proteins perform most of life's functions. If you remember when we learned about enzymes, enzymes control chemical reactions. We said that if enzymes weren't there to control and speed up our chemical reactions would go so slow, life wouldn't even be possible. So since enzymes are a type of protein, they are extremely, extremely important for life. Second, proteins make up a majority of cell structures and body structures as well. Okay, so if you look at all the structures inside of a cell, most of them are made out of proteins, and of course lipids, carbohydrates, but a lot of structures are made out of proteins. Also body structures. Okay, there's proteins that hold our skin together, for example. If you ever heard of collagen, collagen is a protein that we make that holds our skin cells together nice and tight. As we get older, we start to make less and less of that collagen protein, and as a result, our skin starts to sag, we get the wrinkles, all that stuff. So proteins build structures in our cells. Proteins also build structures in our bodies. All right, now I want you to think of this first before I show you. Proteins are made up of units called, so smaller subunits called what? So what do I connect together? Because proteins, they're one of our 4 macromolecules. Remember, proteins, carbohydrates, lipids, nucleic acids. Okay. So for a protein, there's going to be monomers, smaller units that connect together to build the protein. What are those units called? All right, you should have gotten amino acids. Okay, so amino acids are what we're going to connect together to make DNA. I'm sorry, proteins. Don't confuse that. That's often, I'm kind of glad I made that mistake there. Students often confuse what builds DNA with what builds proteins. So DNA, to build DNA, you connect together nucleotides, remember, to build DNA, because it's a nucleic acid. Proteins, a different macromolecule, you connect together amino acids to build proteins. All right, now in order to build your proteins, what makes proteins different has to do with the number and arrangement of amino acids. Okay, so for example, the protein made to cause your eyes to be blue versus proteins made to cause your eyes to be brown, they're built out of different numbers of amino acids and different arrangements of amino acids. There's 20 different amino acids that you could connect together to make proteins. That doesn't mean you can only put 20 in a row. You could put way more than 20. Example here, a chain of 7 amino acids, so if we just took 7 out of 20, can make 1 billion possible combinations. So here's 7 different amino acids here. Okay, so 7 different amino acids, you can make 1 billion possible combinations. So basically, there's almost limitless possibilities as to how we could build these proteins. And when you build proteins with different amino acids, different numbers of amino acids, it causes structures to be built differently. It causes chemical reactions to be, sped up, slowed down, all these different things. Just by changing the arrangement of the amino acids, you make different types of proteins. So there's almost a limitless possibility of what we could do with building proteins. Alright, so the 20 different amino acids, when we learn these, you don't have to memorize them. So don't freak out and think, oh my god, I have to memorize 20 different names of amino acids. Just remember that there's 20 different amino acids you can use to build a protein. When we learn the amino acids, we're only going to learn the beginning letters, the first 3 letters of the amino acids. So here's an example. Here's the 20 different amino acids. Okay, so there's 20 different amino acids that you can connect together to build different proteins. Alright, finally the heart of the notes here. Protein synthesis. Remember, synthesis means to make, and then we're talking about proteins. So the process of making proteins. Protein synthesis, you could think of it as reading the DNA instructions to make a protein. And that's actually what we're going to learn. We're going to learn how to actually read the DNA instructions and build and actually put together a protein. So there's 2 steps to making a protein. So you can't just simply read the instructions and bam, there's a protein. So the first step of making a protein is you have to rewrite the DNA instructions into RNA. And the reason you have to rewrite the instructions is because DNA can't leave the nucleus, and the place where proteins are made is out in the cytoplasm. So DNA doesn't leave the nucleus except when a cell divides, and the place where you make the proteins are out in the cytoplasm. They're the ribosomes. So the DNA instructions have to have a way to get to the ribosomes. So what happens is we rewrite the DNA instructions into RNA. RNA can leave the nucleus and go to a ribosome. So the first step is rewrite the DNA instructions. It's that simple. Just simply take those instructions and rewrite them into RNA. That first step is called transcription. Okay, now in transcription, okay, this is an important part of this word, script. Okay, script is like to write. So with transcription, one way you can remember that's the name of the first step. It means to rewrite. So transcript, right? Scription means to rewrite the DNA instructions into RNA. All right, the second step, you have to now take those DNA instructions that you rewrote into RNA, and you have to read those instructions. And the way we read them is every 3 bases at a time. Every 3 bases is going to tell us something about the protein. So reading the DNA instructions, the RNA instructions, sorry, to build a protein is called translation. Okay, so translation, reading the RNA instructions to make a protein, to actually build the protein. So the flow of instructions is often symbolized like this. You start with DNA, because DNA has instructions for making proteins. The DNA is then rewritten, so those DNA instructions are rewritten into RNA. RNA is going to leave the nucleus, go find a ribosome, and when you read the RNA every 3 bases at a time, you make a protein. So you start with DNA, make RNA, and in the end of protein synthesis, the whole goal of making a protein, you wind up making that protein. Okay, so DNA has the instructions. They have to be rewritten since DNA does not have the opportunity to leave the nucleus. Rewritten into RNA. RNA leaves the nucleus and finds a ribosome. RNA is then read to make a protein. All right, so before we wrap up these notes, let's make sure you can answer these 5 questions here. Okay, so you should be able to answer each of these 5 questions before we move on to the next part of learning about protein synthesis, which is actually the 2 steps and what actually happens. So first, you should be able to answer what a gene is. So what is a gene? Second, what are the units that connect together to make a protein? Third, what is protein synthesis? So how would you describe what protein synthesis is, what it means? Number 4, what are the 2 steps of protein synthesis called? And finally, why are there 2 steps? So why is there 2 steps instead of 1? Why don't we just read the DNA instructions and make a protein? Okay, so you should be able to answer all 5 of these questions. If you can answer all 5 of these questions, which are very important questions to be able to answer, you are ready to learn about those 2 steps, transcription and translation. All right, and those 2 steps will be in the next video.